Welcome to our introductory lesson on cell division. So there are different roles for cell division. There's asexual reproduction and sexual reproduction. Just a quick overview. In asexual reproduction, you have one parent and their offspring is going to be an exact copy. In sexual reproduction, you have two parents and they come together to form an organism that is genetically unique and very different from either parent. So how does this work? If you have a single-celled organism, it's going to reproduce asexually to make another copy. And some of those organisms that we saw under the microscope, like the paramecium and the euglena, they will engage in asexual reproduction to create more copies of themselves. A multicellular organism, like a human, for example, will also get larger through asexual reproduction. Now, growing from a fertilized egg, so each human was once a zygote, and growing from that zygote into a baby, into an adult, that involves asexual reproduction. Asexual reproduction helps replace cells that have died or that have become worn out, like our skin cells are constantly being worn out, and we're constantly replacing them through asexual reproduction. And sexual reproduction, on the other hand, involves the formation of sperm and egg, which come together to form an organism that is very unique, just like every single one of you is extremely unique, and you're not an exact copy of either of your parents. So here's an example, a really good photo of asexual reproduction. You have a paramecium dividing, creating two daughter cells that are now going to go off and be their own separate organism and do their own separate things. And here is a nice picture of a family that shows you the diversity that comes about through sexual reproduction. In this lesson, we're going to focus a lot on asexual reproduction. And prokaryotes reproduce through something known as binary fission. That's their form of asexual reproduction. And you can see bacteria right here engaging in binary fission, which is asexual. And then eukaryotes engage in mitotic cell division, which is more complicated. We're going to look at each of those. So bacteria first and prokaryotic cells first. So prokaryotes have a simple structure. They have a single circular chromosome instead of having many chromosomes. It's not in a nucleus. It's in the nucleoid region of the cell, but it's kind of just hanging out in the cytoplasm. And the other concept we want to introduce here is the genes. So the chromosomes contain genes, and genes code for proteins that actually make you the way you are. And the proteins cause you to have certain features, okay? So this is the prokaryote looks like. How is it going to divide? It's going to divide through something known as binary fission, and I want to show you with pictures what that is. So what really happens is your prokaryote has to make a copy of all of its DNA. All its single circular chromosome gets duplicated. And here you can see the chromosomes have been copied. Here's a copy here and a copy on the right side. And now the cell is going to start elongating, and the copies are going to be pulled apart. You can also see that the plasma membrane is starting to pinch together, and eventually it's going to divide completely, resulting in two daughter cells. Here is an electron micrograph view of the same thing, and then I'm actually going to show you a video that shows you how this happens in E. coli. If you remember, E. coli was in the news recently. It led to this huge recall of romaine lettuce. This is what the E. coli look like when they're reproducing. And they can reproduce quite quickly, not as quickly as this video. This video is obviously sped up, but they can divide once every 20 minutes. I mean, just think about the human life cycle and how many years it takes for a baby to grow into an adult and reach sexual maturity and reproductive age. Bacteria do that in like 20 minutes. So it's pretty crazy what numbers they can get to in a very short period of time. Okay, so when we talk about eukaryotes now, because we have gotten prokaryotes discussed already, eukaryotes are more complicated, and their organizational structure of their DNA is more complicated. Eukaryotes typically have more than one chromosome instead of a single one, so their DNA is in like little segments, and the segments are called chromosomes. So just so you see how this happens, the DNA is actually organized because there's a lot of DNA, and what happens to the DNA is that it's wrapped around these beads over here. The beads are called histones. And as the DNA winds and wraps around the histones, we call that resulting structure a nucleosome. Okay, so this is basically how DNA is organized instead of a eukaryotic cell. We call the DNA together with these proteins, we call that chromatin. Okay, So what happens when the cell wants to divide? Because even though this looks kind of organized, it's not really organized enough. If a cell wants to divide, it really needs to be able to separate out the DNA and work with something that's a lot more condensed and easy to work with. Okay, so usually the chromosomes exist as nucleosomes, but when the cell is getting ready to divide, the first thing it's going to do is make a copy of everything, just like with the prokaryotes. Eukaryotic cells have to make copies too, and then they have to take those copies and their originals and they have to condense them into an easy to work with shape, and that is when you start to see this rod-like shape. This characteristic, instead of this long twisty thing, you see these rods. Okay, think about it as you want to organize your house. If you kept everything loose and separate, it's very difficult to work with and find everything. So we have drawers, we have boxes, we have organizational systems. The cell does too, and it organizes its DNA into boxes known as these condensed chromosomes. Okay. Now, what you have to know about this is that when you look at this X-shaped structure, this chromosome, it's actually composed of two copies, and this side over here is an exact copy of this side over here. They are called sister chromatids, and they're going to be eventually separated from each other, and they're going to go into the separate two new cells that you are forming. They're held together in the middle by a structure known as a centromere. So this is the overall picture of what happens, right? DNA is wrapped around histones, which forms these nucleosomes. The nucleosomes are further coiled and condensed, and then these coils are condensed further to form chromosomes. And these chromosomes are what are going to be used. They only appear right before cell division. Normally, the cell is more in a loose form of chromatin. Okay. Here is a nice picture, an electron micrograph view. Here's a centromere, and you have your two sister chromatids, which again are copies. Here you can actually see it in an actual cell. This is an African blood lily. You can see the condensed chromosomes over here. And then again, this is the big picture. We have our chromosome, we make copies. Copies are held together by the centromere. We call the copies sister chromatids. And then during cell division, they get pulled apart into two separate daughter cells. So what about humans? So humans have their DNA chopped up into really 46 segments, and we call those segments chromosomes. So humans have 46 chromosomes. We're diploid because we have two complete sets of chromosomes. We have 23 from our mom, and we have 23 from our dad, and they come together, they add up to 46, which is the diploid human number. Now, these chromosome pairs are called homologous pairs. You get one from mom and you get one from dad. And the homologous pairs, you can see it over here, are the same length, shape, and they have similar genes. Like if this one has a gene for eye color, then this one also has a gene for eye color. So those are homologous pairs. And all of our chromosomes are in pairs with one exception, or in homologous pairs with one exception, and that is in males. Males have sex chromosomes of X and Y. And those are not homologous. You can see they're not the same size. They don't have the same genes. But the rest of them are all homologous pairs. This picture, by the way, and I'll show you another one, this is known as a karyotype. It's an actual picture of the chromosomes inside of a cell. And this can be used during genetic testing to look for diseases. It can also be used to determine the sex, biological sex. And you can see this one over here has two X chromosomes. This is a human female. Females have no Y chromosome. They have two X chromosomes. Okay, so asexual reproduction in plants and animals and really in eukaryotes, it happens through mitotic cell division known as mitosis. Now, this is used to grow a fertilized egg into an adult, to increase the number of cells in a multicellular organism. It's also used to replace dead cells, and it's also used to help reproduce single-cell eukaryotes. Okay, so how about humans? When do we use this mitosis? When would we use asexual reproduction? And I'm going to show you an overall picture of the human life cycle. We're going to start with the egg and the sperm, and we're going to keep referring back to this picture to make sense of it. So females produce eggs, males produce sperm. Now, egg and sperm are very unique because they don't have 46 chromosomes. They don't have the normal diploid number. They're haploid. They have 23 chromosomes each. They have half the number. And what happens is the egg and the sperm come together through the process of fertilization. The sperm is going to put its DNA into the egg through fertilization, and they're going to put their 23 and 23 together to form a zygote that has 46 chromosomes. So this is the first cell that you ever wore. It was called a zygote. And that zygote is going to go through the process of mitotic cell division, mitosis, and this is asexual, and it's going to start splitting. And the zygote splits in two, and then it splits again, and again, and again, and again, and again. It makes a ball of cells that keep splitting and splitting and splitting and splitting, and this is asexual at this point. After this happens, there's a process known as differentiation. And we learned about differentiation a little bit before, but during differentiation, different parts of that DNA, even though the cells have the same DNA, different parts of the DNA, different genes will be turned on and off to help create all the diverse cells that are in the body. So some cells in that ball of cells are going to become brain cells, some will become digestive system cells, some will become muscle cells, blood cells. So they are going to differentiate until you form a human adult with all these separate organs and organ systems. Females are going to have ovaries where they produce eggs, and men are going to have testicles which form sperm. And these gametes over here, egg and sperm, are formed through the process of meiosis, which we're going to take a look at in a later lesson. So I really hope that was helpful. Next time we're going to take a closer look at mitotic cell division, but I hope that this overview at least allows you to see how cell division works. Here's a picture of the different steps that you will be able to recognize when you learn the steps of mitosis. But just to point out over here, this is how the DNA is normally organized in chromatin. You can't see the individual chromosomes. And then right before cell division, you do end up seeing the chromosomes, which you see on these different in these different cells. So thank you so much for watching.